Alright, hi everyone. This is the second video lecture for the topic cell death by apoptosis. What we're going to do here is talk about the fact that when cells undergo apoptosis, they exhibit a group of fundamental hallmarks that scientists commonly use to determine that cells are in fact dying by apoptosis. To discuss these hallmarks, I'm going to use this image here, which right now is just showing a single, what is supposed to be healthy cell with a nice round nucleus with its DNA inside. If that cell undergoes apoptosis, one of the earliest things that happen is that they will undergo what is called DNA fragmentation, which means basically they will take their long strands of DNA and they will start fragmenting it into smaller fragments. When you look at the DNA that's been fragmented during apoptosis, oftentimes it appears sort of smaller and tighter, almost condensed. And because of that, and by C, I mean if you look at it under a microscope, and because of that, sometimes we refer to this as chromatin condensation. But essentially, it's referring to the process of when cells take the DNA and they start fragmenting it into many smaller fragments. In addition, it turns out that when apoptotic cells are fragmenting their DNA, they will also expel the fragments from the nucleus into their cytosol. And so now I've depicted that here with these black lines, which are supposed to be DNA fragments that are now expelled into the cytosol. In addition, it turns out that researchers will commonly use the presence of cytosolic DNA fragments as a way to determine whether or not cells are undergoing apoptosis. The way this works is you can take a sample of cells, you can lyse them open, and collect what are called cytosolic fractions. What that means is you can basically get rid of the nuclei and collect only the contents that are inside the cytosol, and so you would call that the cytosolic fraction. Then, of course, you can take those fractions and you can subject them to processes, for example, agarose gel electrophoresis. And you can use the agarose gel electrophoresis to determine whether or not there are DNA fragments within these cytosolic fractions. The idea being that only if a cell is undergoing apoptosis should you detect DNA fragments in the cytosol. And so shown here is an agarose gel from an experiment where researchers actually detected DNA fragments in the cytosol as a result of cells undergoing apoptosis. And so what we're looking at is a gel that was loaded with cytosolic fractions from 4 different samples. This sample here, this lane, is supposed to be cytosolic fractions from cells that were not treated with anything to induce apoptosis, so they were healthy, so you don't detect DNA fragments here. But these samples here are cytosolic fractions from cells that were subjected to something to induce apoptosis for either 1, 2, or 3 hours. And what you can see is as time increases and apoptosis progresses, you can detect the accumulation of DNA fragments in the gel, which again came from the cytosol. And so scientists will commonly use the detection of these DNA fragments as a way to determine that cells were in fact undergoing apoptosis. Okay, so DNA fragmentation and chromatin condensation, that is the first hallmark of apoptosis. The second overall hallmark is that cells will also fragment their nucleus, and so now that's what's being shown here. Instead of just one big round nucleus, now it breaks up into multiple smaller sort of nuclear bodies. And then the next hallmark is that cells will actually fragment their entire self. So there's a fragmentation of the cell into multiple distinct what are called apoptotic bodies. So this is a schematic depiction of apoptotic cells, or of the process of apoptosis. Shown on the right is actual light micrographs of both normal cells and an apoptotic cell. So these cells were stained with a purple dye that binds to DNA, and so that's what the purple stuff is. So on the top you can see this nice big round purple stain, which is the nucleus, and which is basically what you would see in a healthy cell. Down here is the apoptotic cell. And as you can see, the DNA is all broken up into these different fragments because the nucleus is fragmented. And if you take a close look, you can make out the apoptotic bodies from the cell also fragmenting. Well, now I want to remind you of something that I told you about in the first video, and that is that a key advantage to cell death by apoptosis is that it entails the removal of cell corpses by phagocytosis, which in turn avoids inflammation. Now keep in mind, this statement here is referring to apoptosis when it takes place in a multicellular organism, right? So that's what this is referring to. If apoptosis happens in a multicellular organism, that organism will have basically cells that will remove the corpses through phagocytosis to avoid inflammation within the organism. And so that's what this image here is trying to depict. Imagine that this is a cell that just died by apoptosis in an organism, and now what's happening is that organism is going to have some phagocytic cell, which is going to internalize those apoptotic bodies, whereupon it will be delivered to its lysosome, broken down, and then its constituents will be recycled. This phagocytic cell is actually the most common phagocytic cells that carry this out are actually a group of cells that are called macrophages. And so macrophages are the cells within us humans, and many other multicellular organisms that basically function to go around our body and identify and phagocytose apoptotic cells. With that said, it raises a question that I want to think about, and that is, okay, well, if macrophages are specialized phagocytic cells that are responsible for internalizing and degrading or breaking down apoptotic cells, how do macrophages manage to identify specifically apoptotic cells as opposed to normal cells, right? Because obviously it wouldn't be advantageous if the macrophages were internalizing and breaking down normal healthy cells. And it turns out that there's a two-part answer to this. The first part of the answer is that it turns out that another key feature or hallmark of apoptosis beyond what is listed here is that cells will externalize something called phosphatidylserine during the process of apoptosis. And so what we need to do is explain what phosphatidylserine is. Well, to do this, I can bring us back to an early lecture where we were talking about molecules of cells, specifically talking about lipids and phospholipids. And so remember we said that there are several different phospholipids that you find within cell membranes, and one of them is phosphatidylserine. And so that's what's shown here. So when we say cells externalize phosphatidylserine during apoptosis, we are talking about cells taking this molecule, phosphatidylserine, and externalizing it, putting it on its outside. In addition, it turns out that healthy cells actually localize phosphatidylserine specifically to the inner leaflet of the cell, at the plasma membrane, that is. And so that's what this image here is trying to show. Imagine this is supposed to be the plasma membrane. Here's the outside of the cell, here's the inside of the cell, and you can see that these are all the different phospholipids that are making up the outer and inner leaflets of the plasma membrane, along with cholesterol. Shown down here this is supposed to represent phosphatidylserine, and note that the image is showing phosphatidylserine specifically localized only to the inner leaflet of the cell. The key is that cells will do this. They'll localize phosphatidylserine to the inner leaflet of the cell as long as the cell is healthy and not undergoing apoptosis. But like we said up here, when a cell undergoes apoptosis, one of the key things that it does is it will externalize the phosphatidylserine. What I mean by that is it's going to take the phosphatidylserine and swap it from the inner leaflet to the outer leaflet, which in turn puts it on the external side of the plasma membrane. Well, the key, and also just want to point out that cells have an enzyme that catalyze phosphatidylserine externalization, and the name of that enzyme is scramblase. And so we're going to come back to scramblase and talk about how this thing is activated during apoptosis to carry out phosphatidylserine exposure. Okay, so the fact that cells externalize phosphatidylserine is the first part of the answer for how macrophages are going to identify apoptotic cells, right? What they're going to recognize is the fact that cells have phosphatidylserine in their outer leaflet of the plasma membrane. And the way that the macrophages are going to do that is it turns out that they express a transmembrane protein at the plasma membrane that we call the phosphatidylserine receptor. And what this receptor does is it recognizes the externalized phosphatidylserine, which in turn leads to the internalization of the apoptotic cell. And so now we can come back to this image and show where the phosphatidylserine is involved. And so now I've sort of uncovered the rest of this image. So at the top here, this is the normal cell that's not undergoing apoptosis, and all these little things here are supposed to represent phosphatidylserine, with it all being what's supposed to be in the inner leaflet of the cell, and therefore all of the phosphatidylserine in contact with the cytosol, none of it on the outside of the cell. However, like we just said, when cells undergo apoptosis, scramblase is going to take the phosphatidylserine and flip it to the other leaflet so it's externalized, which in turn puts it on the outer membrane and makes it accessible to the phosphatidylserine receptor, which you can see this is supposed to be showing the phosphatidylserine receptor on the plasma membrane of the macrophage, recognizing the presence of phosphatidylserine. This is the key or this is the thing that the macrophages recognize that in turn trigger it to internalize or phagocytose the apoptotic cells. All right, and so with that basically concludes this video, right? Talking about the fundamental hallmarks of apoptotic cells: DNA fragmentation and chromatin condensation, fragmentation of the nucleus, fragmentation of the cell into apoptotic bodies, and also the exposure of phosphatidylserine, which is what macrophages use to internalize and break down the apoptotic bodies. Now what we're going to do is move on to the next video and talk about the molecular events that specifically carry out those hallmarks that we just went through.